We are ready to go.
Good morning
Okay,
to
sir.
all. 93 people, 96 people have joined. Good.
7, 98, 99, 100.
Oh, we reached 100.
The century
We are
crossed.
go. The live link has started and the recording is also started.
Okay.
Okay.
Okay, Dinesh sir, so should I start?
Yeah, ma'am, go ahead.
A very good morning to one and all. Our speaker, eminent speaker for today, Dr. C. N. Ramchandran. It's my privilege to introduce him this pleasant morning. Dr. C. N. Ramchand is has done his BSc in chemistry at SD College, Alleppey. He's done his master's in chemistry from University of Kerala. Dr. Ramchand is a PhD in biochemistry from the MS University of Baroda. He has also served as an assistant professor at the MS University. He later obtained his MPhil also from the same university. Dr. Ramchand has also won the Young Scientist Award from Charing Cross and Westminster Medical School, UK, for 3 times for the work in the role of membrane pathology in schizophrenia and the development of EPA as a therapy for schizophrenia. He has extensive experience in basic research activities, having handled prestigious projects in the US and UK, both universities and in the pharma industry. He has over 25 years of rich experience in the areas of teaching, basic research, drug discovery program, and disease mechanisms, as well as guiding MSc and PhD students and setting up research facility. Dr. Ramchand has published over 70 peer-reviewed papers in international journals, edited 5 books, has 3 patented products, and has applied for over 15 patents. He has done over he has over 100 research presentations at international conferences worldwide and was also a senior visiting fellow at University of Sheffield and University of Newcastle upon Tyne, UK, and at Slovakian Academy of Sciences, Slovak Republic. He has a very close research association with State University of New York at Buffalo, USA, Florida International University, and is also an adjunct faculty and PhD guide at the MS University of Baroda. I welcome Dr. C. N. Ramchand and I would request him to deliver his lecture. Thank you.
Good morning, everybody. Can I share the screen? You will have to remove this. Yes. Let me share the screen.
Yes, sir.
Good morning again. Good morning, those who are in India. Overseas, I know that few people are attending from US and Europe too. So good morning to everybody. And thank you, Professor Ramchandran. And our relationship goes to a very long, because I was also teaching in the university, MS University. And from that time onwards, I know Professor Ramchandran. And recently I have been part of the Navrachana University. And that's why he requested me to give a talk on the molecular diagnostics and therapeutics for coronavirus. Let me tell you, I don't think anyone is that expert in COVID-19 or coronavirus, but there will be experts in virology. I'm not an expert in virology too, but my association is connected to therapeutics, drug discovery, and related area. But interestingly, a lot of people got interested in coronavirus recently, and we also got few projects from government, funded projects, and part of our work is connected to coronavirus. In this connection, they have been requested by many people to give talks. So thank you again. And being a Sunday, I think everybody want to take this instead of attending this talk. Thanks a lot. Thank you very much. And I'm having about 44, 45 slides, and let me see that how I can complete. And initially we thought that we will do it at 2 sessions, maybe 45 minutes each. Instead of that, thought that, I will continue. And in between, at any time to stop, please let me know. I can stop and we can continue. Probably it will take about 1 hour, 15 minutes talk. And also want that, what I am involved is high molecular mechanism and related things. So wherever possible, I will try to simplify as much as possible, because I know that a lot of students are attending. What you are seeing here is and various companies that we are involved with. So I will be giving more details about these companies, what we are doing, and how this is related to coronavirus or COVID detection or diagnostics and therapeutics of coronavirus. So some of the companies that I'm involved, I've started and developed are Saxon Life Sciences, mainly involved in novel biologics for macular degeneration. Theragen Biologics for diabetic retinopathy, again novel molecule. Theragen Life Sciences, an ANDA company, abbreviated new drug application. MagGenome is a very successful company, which the technology I have developed using magnetic nanoparticles. We have other companies, Cygenome, then Cygenome Life Sciences, Cygenome Research Foundation. So I'm directly or indirectly involved in many of these companies. We also have a biotech biopharma skill enhancement program, in which what we are doing is that training students. Every year we take about 20 students, train them, because it's very difficult to get students or trained people, especially when we are involved in novel product or technology developments. I'll just briefly give the overview. So the first is COVID-19 detection strategies. What are the ways by which you can detect the virus? Various ways are there. You know about the virus. Virus as RNA, virus as protein, and how these are detected. What is the best methodology? Please remember, this came up suddenly, the pandemic, and everybody has to develop the technology very quickly. But then scientists did that. So RNA-based methodologies. What are the methods available? And I will be briefly talking about that. And I'll be talking about our technology, what we developed. So there are some very interesting technology which we have now commercialized very recently, and we got some funding also, the BIOLOGICS funding. So I'll be talking about that, what exactly we have done. Then there is antibody and antigen detection. How the antibody which is developed in the host or in the patients or in the blood, how this can be detected. That is not the disease detection, whether basically serology, whether it is mainly will tell whether the patient has exposed to virus or not. The other one is antigen detection. Now, let me also tell you at this point that, many of these methods not foolproof. RT-PCR probably is the best. Reverse transcription, polymerase chain reaction, it is also called qRT-PCR, quantitative, or many ways it is not, and probably I will explain what exactly is that. Then therapeutic strategies. So multiple therapeutic strategies are possible. The vaccine, of course, is the best and foolproof. Of course, even most of the vaccines can be effective up to 60, 70, 80%. No vaccines are known to affect 100% of this. It's not possible. But if 50, 60 people that vaccinated, there is something called herd immunity will come. That means those people can protect the other people even they do not have the antibody. So I'll be talking about what are the ways by which various vaccines are developed or antibodies are developed. So, and the other thing is that, drug discovery and development is a very, very, very long process. It takes about 15, 16 years. A pandemic suddenly comes, nobody can predict, and you get 1 or 2 months time. Can the drug be developed? That's why people are using, there is something called drug repurposing, existing drugs. So there are multiple ways to do that one, how this can be done. So these also will be covered. Now, all about coronavirus, I think there is no one in the world now knows, everyone knows what exactly a coronavirus is. I don't think that probably 10 years back, 5 years back, people knew that much, because now any common person knows what exactly is the virus. But still, I will just recap to you what exactly is coronavirus. You know that virus is an extremely small particle or small organism which can't exist itself, because it needs a host cell like a bacteria, or a host cell like mammalian cells is required for its growth and the viral replication process. A large family of viruses affecting animals and humans. So, but let me tell you, most viruses are innocuous, nothing will happen. And the other thing is that if you see, we have about 255 or 56 viruses are there that is involved in making simple cold. Some people may not get cold at all. Some people will get, as you know, rhinitis. Some people will get fever. Some people will get along with that asthma. So various symptoms are there. But please remember that even simple cold or flu, there is a death of 0.1%. So it's not that simple. But then also remember, nobody is involved in diagnosing what type of virus is involved in the flu at this point. And also, there are some flu vaccines are there, but you know, as therapy, only supporting therapy is there. But that is not the case now, because the death rate is very high, especially aged population, when there is comorbidity. That's why if there was another 0.1% death after, probably people may not have been that much bothered, even if it becomes a pandemic, because it ranges from 2% to 50%, depending on the age and comorbidity. That's why everybody is worried. In humans, it causes respiratory infection from simple common cold to severe diseases like SARS, MERS, and recently COVID-19. Let me tell you what exactly is COVID. COVID means corona, CO means corona, VI means virus, D means disease. That's why the name came. 19 means it came in 2019. The SARS, severe acute respiratory syndrome, and Middle Eastern respiratory syndrome, these are all related diseases. Please remember, SARS and MERS, COVID related, but only 70-80% homology. Homology means the viral RNA similarity is only 70-80%. That means reasonably different. These viruses are detected as early as 1930s. Human coronavirus discovered in 1960. Other human coronaviruses are SARS 2003, MERS 2012, and SARS-CoV now 2019. Just see in the right-hand side. So down, it's a very, the structure is like a crown. That's why it is corona, it is called. And just see the structure, very interesting structure. What you are seeing outside is what is called the spike protein. Some proteins are there expressed that comes out. It is not functional protein, it's a structural protein. Sometimes some of the functional protein also can be expressed outside. So any other organism, it contains glycoproteins, certain enzymes, membrane protein, nucleocapsid protein, and main content is ribonucleic acid, RNA. So that is what the constituent, and it's a very small in size, extremely small. The size can be only 125 nanometers. It ranges from 20 nanometers and 80 nanometers. So that is all about the virus. Very interesting small particles or organism that infects other bacteria or the human cells or animal cells. How did the outbreak begin? Now, please remember, there are millions and millions of viruses, which is unknown, so many viruses are there. But why these viruses? And you know, inside our body also many viruses are living. You all know that, you know, there are millions of microorganisms are in your intestine. Huge number. Does you anything happens? Nothing. It is supporting you. The same way, there is millions of viruses are there. But why suddenly some virus, you know, attacks you? Normally, this virus to live, this has to attach with your cell membrane. So cell membrane has got other proteins, as just like a lock and key, the viral spike protein present outside the virus go and attaches like a lock and key and goes inside. Normally, this doesn't happen because, you know, the variation in our protein is huge and insufficient to transmit or, you know, transduce inside the in our cell. Now, if you see in the case of coronavirus, this originally it is present in bat. Bat is known to have about 500 different types of living organisms or viruses there. Now, it is not known whether this virus directly comes from bat or not. Normally, it is not, because we are not in interaction with the humans and bat. That might be one of the reasons. Now, how this attacks the animals? The reason could be, this virus go on changing its structural elements through the mutations in the nucleic acid, in this case RNA. Once go on changing, because various reasons this can happen, spontaneously, external reasons could be there. So once this changes happens, the protein changes. Then what will happen, as I mentioned earlier, this probably will be able to attach with the surface of the cells in the animals. Now, it's not necessary. If you see the homology between any of these animals, what I have shown, and the human proteins are very different. Normally, it's not similar. There might be some homology, but it is insufficient. Then again, when it is in the animals, it can slightly mutate, then can be transmitted. So this is what is happening. So you will never be able to predict when the next pandemic going to come. So we should be prepared for that. These type of variations can happen. I don't need to tell the death, what is happening, or the cases, what is infected worldwide and how many people are recovered. But one good thing is that this is a pandemic. Corona is a pandemic. But if you see MERS or SARS, the death rate was extremely high. MERS was 35%, but luckily that it was controlled. SARS was 10%. That was also reasonably controlled. But in this case, it became a pandemic, but average about 2%, depends on the countries. Probably India has the lowest, one of the lowest death rate, fatality rate. So what are the symptoms of COVID-19? Infectious disease caused by SARS-CoV-2, first confirmed on 17th November 2019 in Wuhan, China. December 2019, the outbreak in China turned to a pandemic. That was the time people came to know about it. Traced as novel strain, beta coronavirus, 70% similarity to SARS-CoV, which I mentioned earlier, 96% similarity to bat coronavirus. So, very interesting, the symptoms. When I mentioned symptoms, this is symptomatic people. Symptomatic people, it can be extremely minor. And what the prediction or people are telling, or from the serology testing, up to 80-90% people do not have any kind of symptoms. That means it probably attacks. They will not get any kind of temperature or cough or none of the symptoms. But once it attacks, and if it is a slow attack, the antibody will be generated. The moment it is antibody generated, then what will happen is that, the further attack will not be there. The immunoglobulin G, IgG and IgM is formed and you are protected. So for some reason, you have a poor innate immunity. I'll tell you at later that what exactly innate immunity and adaptive immunity and how this is connected to the current disease. So if your immunity or innate immunity is poor, what will happen is that, the virus can attack, go into the cell, replicate, and, the person can be attacked. Then depends on the severity, that how the replication takes place. Now, how it is transmitted, really, I think we do not know. But people are telling it can be through nasal or oral droplets, but then there is also now telling that it can come through air. So let us not discuss too much about that. People do not know. But one thing is reasonably clear. A social distancing is good. And if you keep your hand and all the body parts safe with the, sanitization, it is great. So this is the way to prevent. Then immunity boosting. That's something very interesting science. Really nobody knows that, how the immunity can be elevated, increased. But there are various things has been suggested, but there is actual testing other than adaptive immunity and the immunity boosting or antibody development, how your innate immunity can be really tested, because there is no drugs available currently. Now, what are the various types of coronavirus testing? So the main talk is connected to that, how we can test that. So, you know, some virus contains DNA, some virus contains RNA, some virus contains DNA, RNA. So probably one of the best way to detect is that whether that particular sequence. So it's not like simple DNA detection. The specific sequence is available or not. That is what we have to test. So in the current, this thing, it is RT-PCR is used. And if you really see, between 60 to 70 or 80% is the accuracy. That means if you go and test, if you are positive, definitely it is positive. But sometimes, if it is negative, it can be negative, but it is not necessarily that, you know, because, you know, the accuracy is only 70 to 80%. So this is something you have to think about. Still, out of all the methodology, the RT-PCR is the best. Now, if you see antibody detection. So two other ways of detection is, one is RNA in this case, by reverse transcriptase polymerase chain reaction. The second is antibody detection. If antibody has to be detected, you have to go into the blood. There is no other way. So using blood, you will be able to detect antibody. How that is done? Then by antigen detection. Antigen is what? Some of the spike proteins or protein present in the virus. So that can be done either from blood or from the oropharyngeal swabs, which contains the virus particles that can be detected. I'll just briefly tell about our various startup companies. So as I mentioned, we have two biologics company mainly involved in the development of antibodies or Fab molecule, part of the antibody, fraction antigen binding. So based on this, we have the technology how to develop novel antibodies or part of the antibody, and now it is called synthetic biology. So no more animal are not required to be used. No one is using any animals for antibody generation. It's synthetic biology and fast display technology and multiple other ways. Briefly, I will tell about that. As I mentioned, MagGenome is using magnetic nanoparticle. We have developed multiple technology like DNA extraction, RNA extraction, protein extraction, all novel technique, absolutely patented technology. We have published about 30, 40 papers. Here we got few funding from the government. One is the viral transfer medium. So if you know, you need that, you know, oropharyngeal swabs when you take, you need to put it into a medium that is called viral transfer medium. So that medium, a unique medium we developed, which do not require to be kept in the fridge or freezer. That is one thing. Second thing is that a very quick method for RNA extraction. Please remember the entire technology, that's the most important thing. So you can go wrong at 3-4 places when RNA is. That's why you are getting only 60-70% accuracy in testing. If you don't do properly your swab, you will go wrong. Your medium is not right. The virus will not break open. You will not be able to extract the RNA. Your RNA extraction has to be unique, extremely good. Otherwise, you won't get the RNA. Of course, the main thing is that after that, reverse transcription, polymerase chain reaction or RT-PCR. We have another company which is involved in NC, small molecules, AND, abbreviated new drug application, where we have the technology of what is called drug repurposing. Drug repurposing is very, very important. You would have heard so many drugs are coming up. Redmesir or, you know, chlorpromazine, like that. About 4 or 5 molecules are being tested. Now, as I mentioned, 15 years is the time required for developing a new drug. If you want to develop a new drug for corona, forget about it, you have to wait for 15 years. So that strategy will not work here. That's why everyone is using drug repurposing, small molecule. Vaccine, on the other hand, is slightly different. If you want to develop antibody, it will take much more time. So we'll come into that. SIPWISE is another company we are associated with, and we are supporting them in developing various type of drinks. One of the drinks which we are going to introduce is immune booster using certain natural products. So we have also another company called SciGenome and SciGenome Research Foundation, where also we have got some funding, and mainly for antibody antigen detection using lateral flow electrophoretic method. This is basically the MagGenome is all about magnetic nanoparticle. You can see right-hand side is triferric tetroxide. And we are using this technology for all different types of DNA extraction, extremely quick, extremely better than the existing ones, and cost effective. We are also making multiple affinity resins called express affinity RNA extraction kit. We also developed a very good method for total RNA extraction from saliva also. That's another one. Then viral transfer media and few other things are also we are involved in developing. So these are some of the products currently in the market, and the technology is quite interesting. We are the only one using triferric tetroxide. It's very easy to make, and any particle size you can make, but we are using what is called naked nanoparticle. The reason for naked nanoparticle is that every big companies are all using coated particle, but coated particle has got inherent disadvantages. So this is the technology we have developed, and all these things, our technology is used mainly for the mediums, hydrophobic, hydrophilic, and hydrophobic, lipophilic balance is done in order to precipitate DNA, RNA, protein. That's what we are doing, and the various products are developed based on that. Now, our immediate strategy right now, based on coronavirus, is that launching the swab-based RNA extraction kit and unique patented viral transfer medium in July. We have already done that. We have a viral transfer medium already in the market. The RNA extraction just gone for testing for the approval, because all these things has to get the government approval. On the other hand, we are funded for developing a saliva collection based COVID detection kit. So you know that right now there is only three methods: oropharyngeal method, or there is what is called throat, deep throat, in which from do like this and spit over. So that is the way currently used, but in India I think oropharyngeal swab only is. So we are developing this. It's already working, but it will take some time to get the approval. This is the method. Very simple method. You don't need any centrifugation. You take the swab, put it into our medium, then using nanoparticle, you separate it. You will get an extremely pure RNA or DNA. That is what we are doing, and you can test it. And the total time is about 15 to 20 minutes. Patented bare nanoparticles or naked nanoparticles are used. Enzyme and solvent-free workflow. We are not using any of those things. No centrifugation, cost-effective and highly efficient. And COVID detection using total saliva. See, right now, if you want to detect, you have to go to the hospital, because some health care worker has to use the swab in order to take the oropharyngeal swabs. So if once this comes out, this will not be a problem, because you can spit your saliva, you will be able to do. Now we have also detected that, but as I mentioned, it is going to take some time, few months to get the approval, because novel method, because this virus is present in the saliva too. Now what are the advantages? Safe method for sample collection, avoids risk on infection to healthcare workers. Sampling can be done at home or remote locations, avoid need for cold chain shipments and storage, saving a lot of time and cost. That's the main advantage, because we developed this method. If you see the top part, our liquid will be there. You spit saliva over here, then close it, the liquid will come there. Then we can send it. And you don't need to keep it in the fridge or freezer. Now, what is the molecular diagnostic test through RT-PCR? 1 minute. So molecular diagnostic test through RT-PCR. RT-PCR, as I mentioned, reverse transcription polymerase chain reaction. Now, what you are extracting is RNA. So RNA has to be converted into DNA, double-stranded DNA. So that is called reverse transcription. So a particular enzyme is there, and it is reverse transcribed to whatever is the number present in RNA, same number of DNA will be produced. But then that DNA is insufficient to be detected. Then you use polymerase chain reaction, which everyone knows. So this polymerase chain reaction multiplies this. Then you will be able to detect the double-stranded DNA. So the procedure is you take nasopharyngeal or oropharyngeal swab, then collected specimen. Specimen is stored at 2 to 8 degree, currently being used. If anybody uses our technology, that is not required. Then the most important thing is that this RNA extraction, you get purified RNA. Then you will be able to get the test results using specific 3 or 4 genes. Now, interesting thing you need to understand is that 60 or 70 genes are there, and you can really use any of these genes. But most of the people are using RNA-dependent RNA polymerase. That is one gene. There are 3 or 4 other genes are also used. So simultaneously, 3, 4 genes will be amplified and detected. Not one gene, because there can be a possibility something goes wrong. So that is why 3 or 4 different genes. And if you see, probably at different countries, different types of genes might be used. Doesn't matter, the results will be one and same. Detection is positive, active state, or early symptoms of infection. Sensitivity depends on the specimen technique. 72% nasopharyngeal specimen. This is something which is worrying, because 60-70% only is the accuracy. Test specificity is 99%. That means if you are positive, you are getting a positive results, it will be definitely positive. So that is what you need to understand. If you are not positive, does not mean that you do not have, probably there is that 30-40% still possibility is there. So that is something people have to understand. Now, overall, what are this particular slides give various methods for detection or molecular detection. What are the things which is used? You might be able to see very many things. The first is, what is antigen detection? Antigen detection is a protein detection which is present in the COVID virus. This can be detected either from the blood or even from the plasma, or from the nasopharyngeal swabs also it can be detected. So this antigen can be detected after a few days also. Even if the virus die, the protein present can be there. So it might be there, but the accuracy is only 60%. ELISA method and LAF, lateral flow method, is used. Both the methods are used. The second is antibody detection. Again, low accuracy, 60-70%. Antibody detection is what? That you need blood only. There is no other way. Because the advantage of these two methods that immediately, within 5 to 10 minutes, you can detect if you are using a strip. If you are using ELISA, it will take more time. So second is antibody detection. But then the most important thing, more accurate one, is the RT-PCR method. For that, you need to extract the RNA. So the single-stranded RNA has to be extracted. Now, there are various ways. This RNA can be sequenced very easily. The RNA can be sequenced with next generation sequencing. It can be made a cDNA and using nanopore sequencing. Nanopore or Oxford nanopore is a new technology using a specific nanopore or a specific protein they are using for the purpose of sequencing. Very interesting technology, one should know, but it is a time consuming thing, extremely good. And right now they have reached about 99.7% accuracy. Of course, this is real-time RT-PCR. And some other interesting technology is there. After PCR amplification, the DNA can be detected using biomolecular sensors. The other one is that CRISPR-Cas, everybody would have heard about that. There is a company called SEROLOGY. They have developed a methodology that is based on CRISPR-Cas. So these are the different molecular mechanism right now, which is used. RNA detection, multiple ways. Antigen detection, 2 ways. Antibody detection, 2 ways. These are the various things which is used. Oxford nanopore is a sequencing methodology, but there are other advantages for this, various things, especially during the vaccine development, pandemic epidemiology, many of these things can be done. So it is based on the sequencing of the viral RNA. What they have develop, or you can develop a particular consensus sequence, and particular that consensus sequence which is present only in this virus can be detected. So viral epidemiology during outbreak. So probably anywhere you can go and detect. You don't need to bring the samples to your place, and the equipment is extremely small. It's about 5 centimeter long and probably weighing about 30, 40, 50 grams, not more than that. Extremely interesting, and Oxford University has developed this there for the last 10 years, but then they have improved the accuracy. If you want to see whether vaccine efficacy monitoring, whether the vaccine is effective or kill the virus, or the virus is continued to grow, it is that for detection purpose. So mainly generates consensus sequence of COVID from RNA, identifies sequence variants. Sequence variants means specific sequence variants which is present in few RNAs present in the virus can be detected, allows decentralized data sharing for global analysis. And how it works, I have already mentioned. Now, our companies are, I will just briefly tell about, Saxon Life Sciences, mainly involved in new biologics development. We are involved in cloning, expression, lab scale development, pilot development, CGMP development, and which consists of upstream, downstream, analytical, formulation, regulatory. It's a large number of things. And as I mentioned, this is the first time a Fab novel molecule against macular degeneration, which is a protein or Fab protein or an antibody fraction to combat or sequestrate vascular endothelial growth factor, which is growing or expressed in excess in the eye, which causes macular degeneration. The other one is that Theragen Biologics, again a similar company, similar technology. We are developing another Fab molecule that is again for VEGF, but this is for diabetic retinopathy. A similar thing, but these are 2 locations within Chennai. Both are here only. Now, how you can develop Fab for COVID or COVID as a therapeutic agent? Now, you know about vaccine, how vaccine works. So basically, the vaccine is like, generated from virus, various type of virus, which is injected, which I will come detail. Now, it takes some time, but which is very effective. On the other hand, second thing what you can do is that you can develop the antibody against one of the viral proteins, the full antibody. Now, developing a full antibody, there is some problem. You will not be able to tell for 100% sure that it is going to be effective. Suppose you have developed the full antibody and the antigen in question is inside the core of the virus, probably it may not be able to go and attack and, sequestrate that particular protein and kill the virus. May not be possible. On the other hand, the molecule is smaller, where you can see. So a 55 kDa or 28 kDa, this is a single strand, a double strand Fab. Then probably the molecular weight is low. This will be able to go and attach. Now, how these are developed? These are developed using a technology called synthetic biology. You can also use fast display technology. So, now, Fab antibody fragment expression and purification detect epitope region from COVID spike protein, highly advantageous over all antibody therapy. You don't need to use any animals to develop this one. Either you can use fast display technology. If you know the sequences, you can use that sequence, synthesize the DNA, improve further by site-directed mutagenesis. So these are the methods that can be used. One is that, it's very important that you need to have the antibody structure. One is by directly getting the antibody, you will be able to develop that part of the antibody using the technology what I mentioned. That is what it is mentioned over here. On the other hand, if you have the antigen, you can develop using fast display technology the antibody against that, full antibody or part of the antibody. Now, one of the easiest and the best way, probably what are the types of antibody formed, and most effective antibody can be extracted from the human blood itself, for which what you need, the virus is attacked to the human and take the plasma. The plasma protein can be extracted, and this plasma protein, from that, you can use our technology, express affinity technology, the entire antibody can be extracted. Further, if you have the specific antigen for the coronavirus, this antibody can be taken, and once you have that antibody, it can be sequenced and understand the CDR, complementarity determining region, and once you have that one, you will be able to develop a very quick fab. So that is this technology. So briefly, fab molecule can be developed, but these are all time consuming. But in the current scenario, whether it is useful or not, that is a questionable thing, but it takes time. So this is the process by which once you have the fab molecule, which is all the work is done in the lab, then you can do the viral neutralization test. This can be done with the human blood, whether you will be able to see whether the virus is neutralized. Then of course, there is multiple ways of, the characterizing the protein, that is the sequence of the protein, then the biophysical, biochemical characterization of the protein, etc. So you can use animal models for testing also, but it takes several years. I'm just telling that this is one of the methods for against coronavirus people can do. Now we have another company called Theragen Life Sciences, which is mainly involved in small molecules and ANDA, abbreviated new drug applications. So we have the technology for developing novel structures and all, but then again, that will not work in this current situation. So the only way is that drug repurposing. So if you are planning to develop a small molecule against the existing targets of the virus, it's going to take years and years. So it's not possible. You will have to repurpose it. Now, how the drugs and how it can be used, this is quite interesting thing. So whatever I am shown here in various colors, these are different stages of the viral transmission and replication. Now, if you remember, the first thing I mentioned is that if you have a virus needs some kind of compatibility with the human cells. In this case, coronavirus, there is angiotensin converting enzyme, which is present in many of the cell types, especially in your lungs. So this can go and attach there. That's the basic requirement. Once, if you do not attach, the fusion will not take place, it will not go inside. Okay. Now, this can be the role of monoclonal antibodies or vaccines to sequestrate this so that this virus will not go and attach to the cell. So it will not go inside. The second way is that, endocytotic process. Many viruses can go into the cell through endocytosis. So that means even if this process is not happening, sometimes by endocytotic process, depending on the virus. Now, endocytotic process can be inhibited by certain molecules. Chloropromazine. Chloropromazine is a 70-year-old molecule, 70 to 80-year-old molecule. It's a repurposed drug for multiple applications. It starts from back in 70, 80 years back. Originally, it was developed as a drug against people with nausea and vomiting during the pregnancy first trimester. So this was very good. That was acting via a receptor called GABA receptor. Subsequently, it was found that this molecule is very good for allergy, antihistaminic, H1 receptor. The same molecule then immediately at that time repurposed for allergy. Within few years' time, a schizophrenic patient who got allergy, he was treated, his schizophrenia went. Then that last 30, 40 years, this was the best drug for schizophrenia. So it was used as an antipsychotic drug. Even now it is used. About 15, 20 years back, it was identified that, it can inhibit the synthesis of cyclic AMP, in the intestinal cyclic AMP, by inhibiting adenylate cyclase. So it is an inhibitor of adenylate cyclase. By cyclic AMP is inhibited, especially like, the diarrhea, cholera toxin-induced diarrhea, etc. So this has become a drug for certain kind of diarrhea. So again, repurposing. Now this is in the phase 3 trial, chlorpromazine, for inhibiting the endocytotic process. Now, you know about chloroquine and hydroxychloroquine. That is also by a different mechanism inhibit this endocytotic process. Another molecule is that baricitinib. Now, suppose this endocytosis takes place, the virus goes inside the cell. Then it is essential for the virus to work, it has to go inside. Once it goes inside, it will give away its viral RNA. Then this viral RNA within the bodies, the host cells DNA, along with that one, it will start producing, or replication process takes place. The viral protein will be produced. Now, these viral proteins and combined together with the lipid which is from the host cell, combined together will again form the virus. Then finally this comes out, the virus. Once virus comes out, then only it has the opportunity to be attacked by like dendritic cells or macrophages to produce T helper cells, T cytotoxic, followed by B cells antibody. So here you can see the multiple stages of the virus which is entering into the cell and various stages by which drug can be used. Now, if you see, all these drugs are existing drugs. This is what is called repurpose. That's only possible. And but one more thing, let me tell you, nobody knows, because everyone thought initially chloroquine is good. Then in a proper clinical trial, it's shown that it is not that effective. The same is the case with hydroxychloroquine. Nobody knows that it is working. But on the other hand, if you really go and see, the science is extremely good, the right science. In vitro, you are getting efficacy. That's why it is used in clinical trial. So one thing to remember is that whatever you do in vitro, whatever the science, in the clinical setting, it doesn't happen. The molecule is not good. So probably Remdesivir, that antiviral drug. So I will come to that. So that is probably some effect is there. Many people are using that one. So as of now, there is no other really good acting drugs are there. But this also we can't tell for sure unless a long-lasting, very large clinical trials are done. And other drug repurposing is that certain other vaccines, whether it is attenuated virus or killed virus, which can produce various types of antibodies, whether they can go and kill or sequestrate this virus. So that is another technology people are working on. That means different vaccine, already existing vaccine. But of course, there is a lot of vaccine trial for coronavirus, various technologies, and something is going to come out very soon. So what I want to tell you is that drug repurposing is the best method currently to combat COVID, but then still we have to identify the right molecule. Now, immune system booster. So before telling that, because the time is not there, Professor Ramchandran, it is okay, another 10-15 minutes. Hello. 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 Professor Ramchandran. Any of the organizers are there? Okay, let me continue. Now, let me talk about the immune system booster. What are the technology that can be used? Now, people are telling about that your immunity is very important to combat. That is true. But is there a real proven methodology drugs is there? In order for scientists to believe that it should be, again, I'm insisting in a clinical setup. There are a lot of drinks are there and telling that vitamins, minerals, caffeine, inulin, natural, all those things combined together, various drinks are there. Is it really good? We really don't know. Now, before going there, I will tell what exactly is that immunity can be divided into innate immunity. Innate immunity, the first is that, you can prevent, for example, the barrier system. What are the barrier system? Wash your hands so that the virus will not come through. Or, close your mouth so that, that's another way.
Yeah, excuse me, Dr. Ramchand.
Yes, I was trying to...
I was muted. Now the host has unmuted me. Yeah, you can continue. Please do take on 10, no problem. Take your time.
No problem, 15, 20 minutes?
Yeah, sure.
Oh, thanks. Yeah. Okay. So let's first talk about the immunity and innate immunity. And innate, the first that, what something which you can really prevent is that your barrier systems. What are the barrier systems? Barrier system is that, like, closing the mouth or nose or washing the hands, etc. We also have, enzyme-based, chemical-based. If the virus or bacteria goes in your mouth, your saliva to some extent takes care. If it goes to the stomach, then what is happening is that, the acid present in that stomach is also taking place. So these are very good, the natural, which is already present. And some of this can, we can prevent by using that, routine hand washing, etc. Now, but let's say that the virus or bacteria goes inside. That is the time your innate immunity. What are the innate immunity? There are immune cells, leukocytes. For example, basophils, neutrophils, eosinophils, natural killer cells, dendritic cells, macrophages. These type of cells are there. Each one has got different role to play. Each one is supposed to kill something which comes from outside. Now, if they go out of this, there is interaction between some of these leukocytes and T cells. The T cells can have a prolonged innate immunity. It is to some extent adaptive, some extent that innate immunity is there. T helper cells and cytotoxic activity of the T helper cells. So this is the way immediately that, your immunity can be taken care of. Now the question is that, is there something which has been shown to improve this immunity in a clinical setting? But the whole thing is that in vitro setting it is there. So that is not sufficient. You have to give in a clinical setting and see that this really improves. For example, the levels of your innate cells or, immune cells goes up like, 2-fold, 3-fold, 4-fold. After supplements, then you can consider that is this supplement is good, this drug is good. And people are also telling about nutrigenomics. That means interaction of certain nutrients with your gene, which activates certain immune system. So nutrigenomics is another area which is coming up in a big way. So there are many things are there. Now herbal drinks, vitamin, minerals, caffeine, inulin, and etc. Then functional drinks. So we are involved in developing various functional drinks, whether it is sexual wellness, weight management, cognition, immune booster, glycemic index control, anti-hangover. So company in Vadodara and called SIPWISE, but we are doing the development. One of the herbal drink which we are going to come out is very soon, September, which will be rolled over. It consists of a couple of natural products, which is otherwise in vitro shown to have very effect in immune booster. So I have worked on curcumin for a long time. I have multiple patents are there. There are 11,000 various applications has been reported in literature, if you see last 40, 50 years, but it is yet to be proved beyond doubt. That's very important thing. And curcumin assays do not have bioavailability. Unfortunately, that's the problem with many of the natural products. If you really see, many has got 0.1% to 1%. Of course, some has got a very good bioavailability also. That is some of the things which is sorted out. Here there is a technology called reverse pharmacology approach can be done. So that I think I'm not able to tell right now because, it won't take, insufficient time. So what I just want to tell you is that one of the immune booster drinks, which we are hoping to introduce within the next 1 month's time. This can be in as small shots, or 25 ml, 50 ml, or as 100 ml drinks. So this is what we are doing, and the mechanism also we have already studied about that. Another interesting, which is a collaboration with the US company, we are involved in developing, called immunoglobulin Y. Immunoglobulin Y they have developed, that is yolk antibody. The chicken can produce antibody, and that will be also present in the egg yolk in high concentration. So we are developing this egg yolk antibody against multiple coronaviral antigens. So, of course, if this is not for injection purpose or a therapeutic purpose, then again it takes years and years. But on the other hand, the egg yolk will be considered semi-purified as a protein source and will make a drink that can be used as a drink, but this will wash out your throat virus or viral particles can be removed. The second thing is that you can use as a spray, throat spray, nasal spray. So this is under development, and very interesting technology, the basic technology. So will be coming out hopefully very soon. So we'll call it anti-COVID mouthwash. The whole thing is that if this is not done within a couple of months, it all won't have any value, but the same technology can be used for subsequent many other applications. Now, some other technologies I'll briefly mention. Some people are using the Raman spectroscopy. Everyone knows about what is the application of Raman spectroscopy. Raman spectroscopy is mainly used for the structural identification of complex structures. And how does it work? Electromagnetic radiation. Electromagnetic radiation can be like, diffraction, reflection, refraction, all those things will happen electromagnetic radiation. So one of the diffraction mode of Raman spectra is that at the electromagnetic radiation, if the molecular structure changes based on the type of molecule or atom, that will give a spectral pattern. You can see. So let's say that a particular sequence of RNA gives a particular pattern like this. Now another viral RNA, each RNA will give a particular pattern. So you might be getting a different types of variants of the spectral pattern, and each one will be having very, very different one. So what is required to do is that, an algorithm has to be made. Algorithm means various virus has to be taken and identify the Raman spectra pattern. Then the COVID viral RNA, that you can take the pattern and see. Then what you can do is that you can take the saliva and check the pattern, and you will be able to see whether there is a COVID virus is there. Probably if it is reasonably accurately, it can be detected. Is going to be a very, very quick method. And probably within few years time or few months time, this technology will come using artificial intelligence. Now you all might have heard about the serology testing. What exactly is that? Now immediately after any foreign material goes inside, the first thing which is happening is that IgM, which is 4 to 5 days, and probably 10, 11 days, it will reach a peak. So its role is that immediately getting activated and killing the virus. Subsequently, within few days, it will start from 15 days onwards, immunoglobulin G will be activated. So if you get a pattern like this, that means both IgM and IgG, the person has recently infected with the virus or the antigen or bacteria or whatever it is. You will be able to get, this need not be this is not a specific antibody. It's a general antibody detection. Now, what will happen is that, within few days, the IgM will not be there. Then, if there is a second attack is there, or at some point after 20, 30 days, the IgG will be activated and it will remain high. That means if you are getting after 3 months, 6 months or 1 year, IgG is high for a particular antigen, that means you already had this attack. Is not the first time, it is the second time. So right now, the pattern could be this or this, and basically they are testing IgG and IgM, whether the person is exposed. Now, one interesting statistics, I heard that Ahmedabad has got 50% people are exposed, probably now by 60%. That means herd immunity will be there. In 5, 6, 7 days, the sero testing in Delhi has shown that it has got about 25%, and by now it is about 35%. That's why there also it is coming down. Chennai is about 25-30%. So what is herd immunity? Now, if you really see what we are seeing, the numbers which I mentioned is only tested numbers. But 95 or 98% people are not tested. They don't even know it is there. Only by sero testing you will know whether the person is exposed. If it is exposed, they never showed it. It's a great thing, because if the immunity 50, 55, 60%, you can be rest assured that it is going to go away. Because there's something called herd immunity. If your surrounding people are immunized, you are not going to get, because you can't get it from that person. So normally about 60, 70% immunity comes. 50, 55 onwards can start declining. Then 60, 70% that you're fine. Very rarely that the pandemic will come back, especially the second or third attack of that particular disease. So this is all about IgG and IgM, and the testing methodology is there. Now, I mentioned very clearly about what is the RT-PCR methodology for detection of RNA. That means nucleic acid detection. But then the other methods are lateral flow assay for the detection of antigen or antibody. So two different things can be done. The lateral flow assay or ELISA test. Lateral flow assay is a very quick method. You will be having a strip. You will be putting the blood into one slot. And so the technology is interesting. If you want to detect antibody, the antigen, the COVID antigen, which will be immobilized in gold particle, which will be in yellow color. If the antibody is there, once it start moving, take the antigen, come over here. Now what will happen is that if the antibody is IgM is there, so what will then is that an IgM antibody will be immobilized here. IgG antibody will be immobilized here. So what will happen? You will get a line, gold line will be here, here. Now, if nothing is there, it will come here, you will see a line here. So this is the way it is detected. So as I mentioned, from blood, you can detect antigen or antibody. ELISA test, I don't think I need to explain. Everyone knows about the ELISA test based on antigen or based on antibody. It's a colorimetric method which is routinely used. But it takes probably a couple of hours, not very quick. But here, at the point of care, you will be able to detect this. So the same way, antigen-based detection, which is exactly what I was saying. So both antigen and antibody, as I mentioned, 60 or 50, 60, 70% is the accuracy, but still as to exactly, definitely it is not 100%. So probably a combinations of tests will give. For example, the antigen testing as well as RT-PCR and the antibody, maybe after 8 or 10 days, will be able to test very clearly whether the person has got me. I'm not sure, but I'm sure 90% certainty you might be able to get. Now, there was a couple of days back, I was giving a talk and I was asked one question, like after 1 month or 2 months, there is again that person is detected using RT-PCR or the antigen testing. Let me tell you, if you already has exposed to a virus and you are cured, the antibody is developed, unless the antigen, unless the virus mutates and a new virus comes, especially in that particular region, what I was telling that, the translocation takes place, very unlikely, but there is no data. But then I'm just scientifically telling. But then what's happening? Now the virus will die because virus replication ability dies once the antibody is developed. After 10 days or 15 days is finished. But still viral particle will be there in your nose, in your blood. So it will take probably few months to metabolize and excrete and remove from your body. So viral antigens can be there. The viral capsid proteins can be there. And these all can be detected by using, whether it is RT-PCR, if the RNA, what is present in the viral particle, it is not metabolized, you will be able to detect that. So I'm not categorically telling the repeat infection, because many people are telling they are getting second time, third time, and it may not be. Theoretically, once you have got the attack, you have developed the antibody, chances are, at least depending on 3 months later, it could be very unlikely. Now, what are the different vaccines under development? Various types of vaccines are developed. So the most popular one, easy ones are inactivated and weakened virus. What is inactivated or weakened virus? Inactivated means like the virus can be killed because virus will be grown in human cells or bacteria. The viral will be extracted. There are multiple ways of killing. For example, formaldehyde is a method which will kill the virus, and even if virus goes inside, nothing will happen. It will not replicate. But the advantage is that this virus can still enter the cells. And what will happen is that, this can produce probably the replication process, or, or at least what will happen is that, this virus, if this large amount is given, if is it unable to replicate, the virus itself has got its own protein. That protein will become antigen and antibody will be produced. See, these antibodies will be polyclonal, and various antibodies for various proteins will be available. What is weak one? For example, this virus can be developed using killing certain certain part of the virus so that it will not be toxic to the host cell. So that is the weakening methodology. So that is by a cloning methodology, you will be able to remove a certain part of the virus. So the weakened virus or inactivated virus. Then what is viral vector? There is something called virosome. Virosome is that the interior part is removed. If you want to put anything inside the virus, you can do that. Now, viral vector also is something like that, in which what you do is that, you can put the DNA or RNA or the protein inside. Then the virus will go, this will release the protein. If it is DNA, that will get incorporated with the DNA system of the host cell and produce the antigen, which will produce antibody after interacting with macrophages. So normally viral vectors are non-replicating. Now nucleic acid, by the way, viral vector, this is successful, and animal vaccines are there. This is the most successful inactivated or weakened one. That's what most of the countries in India especially doing. Nucleic acid, what exactly is that? The DNA or RNA that represents the protein of interest, which is antigenic, which needs to be produced antibody, then you can introduce in the host cell. The introduction is quite cumbersome, but then once it is introduced, this will transcribe and translate and produce the antigen and produce the antibody, but not yet in the clinical stage, at the research stage. Of course, this is the best methodology, protein-based. Protein subunits, you can produce an antigen and inject the antigen, you will get the antibody, and extremely good methodology, and virus-like particles. So what you can do is that, multiple proteins which is present in the viral cells or virus-like particles can be developed and can be injected. So this is the number of vaccines under development. Probably about 110 different vaccines are there. If you see, protein-based is probably the maximum number, about 25, 30 are there. So nucleic acid is there, viral vector 1 is there, and the actual virus also is there. So about 100, 110. So let's all hope that some vaccine is going to come out very soon. How does the viral vaccine work? So this is weakened virus, inactivated virus, which will go inside. As I mentioned, replicate, viral protein will be produced. Once it comes out, it will be interacting with the antigen presenting cell. Antigen presenting cells interacts with the B cells, T cells, B plasma cells, and B memory cells will be produced, and you will get the antibody. Viral vector, something similar to that. So spike gene or spike proteins gene will be introduced. Then again it comes out, the protein will be produced, exactly a similar mechanism, the antibody response will be there. Nucleic acid vaccines, probably, which I mentioned, the nucleic acid will be introduced into the body, but there is using plasmid. So if you see here, this is the DNA of interest that will be introduced in the plasmid, which will be introduced into the cell type. Then you will be able to produce the spike protein, and antibody will be produced. The other one is that you can introduce spike protein or M protein, then exactly the same. So various methods are there. So I just wanted to tell whether the whole virus activated, or inactivated or killed virus can be used. Nucleic acid can be used, RNA can be used, subunit protein-like particles. So various methods are there. Only thing, everything takes time to develop. But luckily, if you see the whole over the world, within 3 months, 4 months, extremely good vaccines have come. Some of the vaccines which is produced, it took almost 30 years. Now, plus seeing 3 months or 4 months, some vaccines are coming. What? Because the regulatory people are also supporting that. If regulatory support is there, really that, the drug discovery can reduce from 15 years to 10 years or even less. Because they are always worried whether it will have some adverse effect. So these are the major vaccine under development. So probably, as I mentioned, 110 vaccines are there. In India also, couple of vaccines are in the development. Hopefully we will be all getting. I don't think I need to tell too much about this. Everyone knows about this because it's a regular news. And you heard about what is convalescent plasma therapy. So in convalescent plasma therapy, a person who is attacked by the virus has got the antibody. After about 15-20 days, he will be having sufficient antibody. You can take up to 400 ml of blood, remove the plasma, introduce the plasma. So there are some adverse effects, and I'm not very sure, but it's reasonably, I think it is working. But scientifically, it's an extremely good methodology. The second thing is that one of the technology, what we are planning to develop is that purify all the antibody, whether it is IgG, IgM, using our technology. We have a one-step technology that can be given. The third is that, only extract the antibody which is responsible for the viral antigen can be taken out. That can be done. So we have the technology. We are planning to work on that one. So this is what convalescent plasma therapy. At present, the people who are infected, they will test using viral neutralization and take the plasma and give it to the acutely, the people who have severe problem. So what is herd immunity? So when it is no immunity, so probably this is mentioning about the innate immunity, high innate immunity. The people can get very easily get that one. Probably, few people will be spared from that. When some immunity is there, much less people. But in herd immunity, if you see, so for example, these people are not infected with the virus, but it is surrounded by people who are infected, then there is no chance for them to get. What exactly is herd immunity is that when it is reaching about 55%, there is a high chance that it will start declining. When it is almost 70 or it is 70%, then chances of getting the other 30% is extremely low. So probably it looks like India might be reaching herd immunity after a few months. So now how to prepare the world for future pandemic, improve hygiene practices, telemedicine, timely identification, efficient diagnosis, rapid isolation and clinical management, strengthen health system, invest in research and development, cost-effective treatment, readily available protocol to be followed during pandemic. So I think with that, thank you very much. Thank you, Professor Ramchandran, giving the opportunity. I think I took almost 1 hour, 20 minutes, but I'm happy that I finished in time. You can come out with any questions.
Yeah, good afternoon, Dr. Ramchand. On behalf of all the participants, on behalf of School of Science, Navrachana University, thanks a lot for an enlightening and a very lucid lecture that you delivered. The presentation is, excellent. It offers a lot of things for us teachers to learn and how we can communicate with the students the very difficult, intricate concepts in science. Thanks a lot, sir. There are a lot of questions and I don't know how I deal with them. Hopefully we will be able to address all of them because there are many. Okay, and hope the time permits and the technology permits us because we have already a lot of time into the session. So the first question, sir, is from Ms. Tanseem from Ms. Tasneem from Inter-University Center for Genomic Research and Gene Technology, Kerala University, and she wants to know about the viral transport medium.
Right.
So what is the viral transport medium? That's the question.
So viral transport medium is a medium which is isotonic in nature, which will have some component to break the cells. It is something similar to your cell culture medium, but there is some additional components are there. So that is what we have developed. It will contain a buffer. It will contain, for example, nutrients will be there. So basically what we are looking is that the viral should be intact. The bacteria should not grow. So when you are taking the swab, it contains the virus, the bacteria, and your host cells also will be there. So it's basically meant for virus. So something what we developed is something unique. You don't need to keep it in the fridge.
Oh, thanks a lot, sir. There's second question from Dr. Sumit Bhaval. His question is, can functional gold nanoparticles be used for rapid detection using colorimetric procedure? Is there a possibility?
Yes. You are talking, whether it is lateral flow detection or ELISA technology, or if it is nothing is there, you can use magnetic nanoparticle. Magnetic nanoparticle, if you're binding an antigen or antibody is easy, you can use for colorimetric method, even lateral flow method or ELISA. So all the three can be used. It can simplify the method. So gold nanoparticle can be used. But you should know how to bind your protein of interest, which can be antigen or antibody. That's important.
There are a couple of other questions related to this magnetic nanoparticle slide that you used during the presentation. There's a question from Dr. Amit Patwa, again a faculty in chemistry at Navrachana University. It is regarding the magnetic nanoparticles that are used for detection. The question is, how stable these naked nanoparticles are? Can we store these nanoparticles for longer time, and are they pH sensitive?
Right. It is, if you are making the particle and putting it into a simple buffer, there can be problem, further oxidation and brown color can come. So we have stabilized that. So at least up to 5 years, it is very stable. That's why we are, so there is no issues. You will be able to stabilize. If you are making that one and you can work on that, or if there is a problem, let us know. We'll tell how you can stabilize.
Okay.
You can write to one of our scientists and we'll tell the methodology. We'll be happy to do that.
Okay, thanks, sir. That will be great. There's another question from Ms. Tasneem again. the viral titer will be low in saliva. So how the sensitivity can be increased in this case, and also how the viral RNA would get attached to the bare magnetic nanoparticles?
Right. The first thing is that, about the sensitivity you asked, isn't it?
Yeah, the question is related to the limit of detection or the sensitivity.
Correct. In the saliva.
Yeah.
Now, so we have already isolated and standardized method for from the saliva how RNA has to be extracted, and we are getting extremely good yield, whether it is RNA or DNA or host cell DNA, host cell RNA or total RNA. So that method is there. Already we standardized. We have already patented, and this publication will come very soon. That is first thing. Now, how this is binding to nanoparticle. Okay. Now, see, when you are at a specific condition, we are making sure that the RNA is precipitated compared to other nucleic acid. That is the first thing. So that is the technology we are using. So when we are adding our specific solution, many of the other product will not get precipitated. Mainly RNA will get precipitated. But let me tell you, when we are doing this, not only RNA, a little bit of DNA also comes. But then that DNA has to be removed using DNase. So that's what we are doing. So then you will get pure RNA. And what we are getting is pure 99% pure RNA.
Thank you, sir. This is another question from Dr. Sangeeta. She is from the Oxford College of Science. She asked that in case of animals like bat and pangolin, the receptor used by virus is also ACE2, ACE2. How or what studies enable us to find the intermediate host, like in SARS? So what are the studies that enable us to find the intermediate host? That is the question.
Now, one is that what you need to do is that, what is the spike protein sequence can be done, followed by the sequence of the angiotensin converting enzyme, which is present in various animals, can be studied. Then you don't need to, once you have the sequence, the DNA sequence, you will be able to predict the protein structure. From the predicted protein structure, you can do a docking study. So this can very clearly tell, you don't have to go and do all the experiments. It's very complicated experiments. So all can be done in silico. I hope you understood what I'm telling. So you need to know the sequence, predict the protein structure from all the 3 species and do a docking study.
Okay. There's another question regarding the kits. Do the kits address variable and very low viral loads at different times or species during the course of infection? That is again regarding the limit of detection of the kit.
Right. We have tested about 200 samples now. COVID, all 200 we compared with Coagen, and we have got even extremely small quantity otherwise detected in Coagen, we could detect. And we have found that the methodology what we developed is about 30-40% better in terms of the quantity and quality is concerned. There is no inhibition, the reverse transcription or polymerase chain reaction.
So there is another question regarding this separation of the viral RNA. Please explain how viral RNA attaches to the appender filter, while host RNA, protein, and other debris come down the filter. So that is regarding the extraction.
That's not the method we are using.
Okay.
No. We are using nanoparticle. We are not using any filter.
Okay.
Not the filtration technique at all. Filtration.
So there is no filtration in the process altogether.
No, we are not using. We are using nanoparticle, and it's a non-specific binding to nanoparticle surface, hydrophobicity, hydrophilicity, electrostatic interaction. This is what we are.
Okay.
So our technology is mainly this precipitation technology, how to precipitate. So that liquid what we are using, a combination of liquid, hydrophobic, hydrophilic, That's the important thing. This question everyone asks.
Okay, yeah. This was again from Ms. Tasneem from Kerala. There's another question. Can the mouthwash antibody against SARS virus neutralize the throat swab attached viruses or the endocytosed viruses?
I'm not sure about, see, this is first of all, we'll have to test it. Okay. This is a very exploratory stage. We are going to get the antibody. So the first thing is that we need to in vitro test and see whether it is really binding. Then we will have to test in the real condition. Then only our proof of concept will come. Now, endocytosed one, I doubt whether it will, no way. Because we are only using in the surface. No, I don't think so. Theoretically not possible.
Yeah. There's another question from Mr. Sanjay Jadhav. I think you have partly addressed is regarding what is plasma therapy. But the second part of question you may address is what kind of plasma is required, and is there any connection with the hemoglobin level with respect to this therapy?
No, first of all, I don't think doctors will take the hemoglobin, or you have some issues. That one, I don't think so. That is one thing. But then anyway, if plasma is taken, the other cell, RBC and everything will be given back. Probably platelet also will be given back. Platelet and RBC. So as soon as you get the blood, this is what I think you will take: plasma, platelet, and RBC. So the other things will be given back. Only plasma will be used. Then, the group compatibility should be there before using. You can't use anybody's blood to anyone. So group compatibility has to be there. AB, A, O, O plus, those type of things. Yeah. That's group compatible.
Yeah, there's another interesting observation from Ms. Nasreen Najib, that why the American people are severely affected than Asians in general, or more specifically Indians, with this particular disease.
Right. Very, very interesting question. I'll tell you, have you heard that, for example, the children in Europe and US, the childhood asthma level is about 20-25%. Very high. You know why? That is because of the clean environment. There is something called Th1, Th2, the T helper cell ratio is involved because of the clean environment. If you see their antibody levels for various antigens and other things, it's very poor. Have you also noticed that many of the Europeans come in India, they are maybe eating very hot food, good water, but 90% people first time get diarrhea. 90%. Some people get severe, some people gastrointestinal problem. Why? That's because they were not exposed to all these, antigens. I always tell, I have never got a diarrhea or, severe gastrointestinal problem because I used to drink simple water from the well when I was young. Any food is eaten. Of course, lately, when I was in Baroda, I only started drinking boiled water. I don't think I have ever taken boiled water before that. So I'm not telling about me, many people it is like that. So you can take any of these food. If you are exposed to all these antigens, virus in a small way, so immunity goes up, mainly antibody. So normally these things will not affect you. What people are thinking is that, exactly the same thing. Overall, their innate immunity is low because, exposure of outside environmental virus bacteria. This is only a theory. I'm not sure whether it is the right thing, but this is, of course, this is what is happening. The death rate is high. The infectious rate is very high. So in US and European countries comparatively. For example, in Hong Kong and Taiwan and Singapore, very low.
Okay. So there are 3-4 more questions. Can we move forward?
100%, yes.
Okay, thanks a lot, sir. There's a question from Dr. Namrata Erande. She mentions that CCMB research recently talked about dry swab and RT-PCR without RNA isolation.
Right.
whereas and using only heating swab in some buffer at 95 degree. What are your views on the same?
See, if they have done, probably it might be working. Yes. So if you really see why you want to purify RNA or DNA and remove the protein, the first question can be asked. You can take the fluid because what you are going to use is very highly specific, whether it is RT-PCR, the nucleic acid sequences. So it is highly specific. Theoretically not required. But then normally what is happening is that, other inhibitors are there. Now, we have also checked without purifying. Normally what will happen, you can get very high false positive, first of all. The amplification will not happen. Theoretically, it should work. Practically, many inhibitors which inhibits the enzyme will be there. So, but probably they might have standardized, or they would have standardized in such a way that, high quantity of polymerase enzyme might be using. So I do not know.
Okay, I hope that answers Dr. Namrata's question. There's a question from Mr. Gangopadhyay from National Institute of Virology. Will antibodies from one SARS-CoV-2 subtype infected patient be able to neutralize a different subtype virus infected patient? If not, should the plasma that is harvested be subtype specific?
See, if you're talking about the plasma therapy, somebody who has got viral attack, what will happen is that it's not one antibody, because this SARS-CoV probably 60, 70 proteins are there. So you will be able to detect all these proteins. Now, as long as the surface protein antibody is there, I'm sure even if one or two antibodies are different, but few other proteins might be sufficient to neutralize. Anyway, the neutralization assay can be done in this case.
So there's another question from Ms. Arshnaz Sayed from Biochemistry Department, MS University of Baroda. RT-PCR, CRISPR-Cas and NANOPORE target sequencing would cost a lot for detection. Is there any accurate and cost-effective technique?
Yeah, you're right. NANOPORE is expensive. RT-PCR, the price can be brought down. Now, if RNA extraction is not required, as you mentioned earlier, probably you take the saliva and you amplify directly and test with the reverse transcription of the methodology. Probably the cost can be brought down. But then please remember, see, the companies that they are making, people might think they are charging 2,000, this thing. But the back of that, there's a lot of work is there. The expenses of research, development, and many failed methodology. So all this will cost. So that's why even many companies won't be able to reach by reducing the price break even. So that's why it is maybe enormous amount can be produced at a later period. Then if you see a lot of competition also is there. Many people are reducing the price also. Eventually it will come down.
Okay. Sir, another question regarding the delivery of nucleic acids. How do we deliver the nucleic acids? Are there any nano strategies for the same?
Electroporation, that is one. The other one is that, nanoemulsification. The other one is that, liposomal delivery. So many people, then virosomes is another one. Virosomes is that the virus coat protein and other nucleic acid, removed everything from inside, and you can put it inside, then the virus can be transferred. That's a methodology which is used. So that's a very successful methodology. That is one, virosome. Then nanoemulsification, liposomes, electroporation.
Okay. So there are a couple of questions from Keval Parmar. He is a first year BSc Life Science student from Navrachana University. The first question is, what are the long-term consequences for those who have survived COVID-19 infection? And the second question is, the world and all of us are constantly evolving. If the COVID-19 infection drops or the pandemic gets over, is there a possibility of major or minor evolution tracks in human or the world?
I doubt very much that human, because in our lifetime or next 1,000 years, anything going to happen just because of this pandemic. Pandemics will come and what is that? You just remember about think about the 2018 Spanish flu pandemic. 50 million people out of 1,500 million people at that time. 500 people were infected, 10% of them died. Anything happened? People all forgot. The same way, this will all come to a normal situation after 6 months. I am very, very sure about it. Please also remember one more thing. Let me tell you. You know how many people die in a day in the world? 150,000. In India, about 35,000 people are dying every day, single day. The death is in coronavirus is 1%. Now, what is happening is that people are also telling because of comorbidity. Now, this 30,000 people, at least that, if they are dying in a hospital, which normally at least 10% might be dying in the hospital, they will be tested for coronavirus. Now, if you remember that, many people as asymptomatic has reached that, they will be either antibody or coronavirus might be there. Now, this will complicate the statistics. That's what I feel. Really, this 300, 400 people, really because of coronavirus or something else, nobody knows. Yes, they had coronavirus, and there is definitely that those who had got severe, problem with this thing. But when there is a comorbidity like the heart problem, then naturally there can be mortality, morbidity, etc. So I don't think we are not going to evolve or change, the mutation going to take place.
Okay.
That's what
Sir,
my assumption.
Okay, yeah. Sir, almost all the questions are done. There may be couple of more questions that we will be collecting from the students, and I hope we can redirect to you, and later on those answers can be sought.
Sure.
Okay?
Yes.
Yeah. So I think from my end, the question and answer session is over. Dinesh sir, can you unmute Professor Ramchandran?
Right now, Professor Ramchandran is not in the
Okay.
attendees
Professor
list.
Yagnik has something to mention.
Yeah, you are.
Please unmute, sir. Yeah. Yes, sir. Go ahead.
Thank you very much, Dr. Ramchandran. Thank you very much, sir, for your wonderful session today. And it was very informative. It was like a popular science lecture. So people, the general public also, as well as people specializing in this research, will find it very interesting. And I'm so happy to see in the audience, there are people from all over the world who are watching right now live. We have people even from Las Vegas in the United States. It's nearing midnight over there, but they are all watching very closely. So it shows the level of interest in your talk. And I thank you very much on behalf of Navrachana University, sir. Thank you so much for enlightening all of us today. And thank you all
very
the
much.
organizers also.
Ramchandran, sir.
Yeah. I'm sorry, I got disconnected in between and I had to change my connectivity. So I missed in between something.
Yes, sir.
So I would like to thank Dr. Ramchand for having accepted our invitation and give a talk on the modern current problem that is going on. And Dr. Ramchand is always associated with us. He is also in the advisory board of biomedical science. And I'm sure he will be interacting with us quite more and certain other things we want to work with him along with our life science section. And he's a very good friend of mine, and I'm sure he will definitely be collaborating with our university and our School of Science. and hope to have more interactions with him in due course of time. And thank you very much. And I think you took your time and then answered all the questions. That's a great thing. And I think also congratulate you for, you have so many companies, you're a classic example of a life science bio entrepreneur. And you're doing wonderful things, developing these, especially these nanoparticle-based extraction techniques, which I think our university will be definitely purchasing for our studies also gradually.
Thank you very much. And especially I am extremely happy to work with anybody in Baroda, because I was really happy to hear somebody ask question from MS University, Baroda Biochemistry. That is my alumni, my department. I did PhD with Professor Ramakrishnan, and I always like to come there. All biochemistry, microbiology, and now your university. And this same week, I have a talk to be given at Parul University. So I don't know, most of the Baroda people are calling me. Thanks a lot. My special thanks to Professor Ramchandran and all others. So hope to see you very soon in person, not virtually. Yep. So looking forward to that.
Okay, thank you very much.
Thanks a lot, sir. With that, we come to an end to this interesting session. Thanks, Professor Dr. Ramchand, once again. Thank you, Professor Ramchandran. Thank you, Professor Nilay Yagnik. Special thanks to Mr. Dinesh Panchal, who is behind all these technical arrangements, which we could do seamlessly. Thanks a lot to all the colleagues at School of Science for taking such initiatives. Thank you very much.
If you have the recording, please send me some time.
Yeah, we have recorded, sir. Mr. Panchal would be doing the next task of having this recording properly arranged, and we'll definitely be sending to you. Thank you, sir.
Thank you. Thank you.
Sure,
Thanks.
sir.
Thank you.
Okay, bye.